Hi everyone, my name is Nivedita Rambam and I will be presenting my study on the topic Relapse in AML post-allogenic-hematopoietic cell transplantation and intensive chemotherapy, a transcriptomics study. So I have completed my bachelor's and master's from MIT University Noida, and I have also worked as a research fellow at Pine Biotech. This project was carried out under the guidance of Dr. Raghavendran L. So in order to understand the study, we need to understand what AML is. So, AML is an aggressive form of blood cancer that takes up 1% of all cancer types. Leukemia, as we know, is blood cancer. Acute means rapid. So, acute myeloid leukemia is a rapidly progressive blood cancer that affects the myeloid cells. So, what are myeloid cells? During the hematopoietic stem cell lineage, the multipotent hematopoietic stem cell differentiates into lymphoid stem cells and myeloid stem cells. The lymphoid stem cells differentiates further into NK cells, T and B lymphocytes. The myeloid stem cells differentiates into RBCs, platelet, myeloblasts, and mast cells in the bone marrow. So when this myeloid cells count goes beyond 20% or more in the bone marrow, or if it goes beyond 5% in the blood, AML can be diagnosed in most cases. Now then, let's take a look at what the problem is and what the purpose of the study is. So, recent studies for the treatment of AML shows that allo-HCT relapse had a high 4-year survival rate in comparison to chemotherapy alone or a combination of these two. So, we'd like to dive into what makes the difference in the survival at the pathway and the genes associated with it. We would also like to study the regulatory link between AML and neurological autoimmune disorders. And we have obtained the dataset from the GEO database of NCBI under the number GSE 197417, and the expression profiling was through high throughput sequencing. From the original paper, they observed an epigenetic marker, PRC2, to be the cause of post-transplantational relapse, and a certain pathway antigen processing and presentation alteration was deemed as one of the main drivers of allo-HCT transplantation relapse as well. Coming to the study pipeline, we first collected the raw data from the GEO NCBI database. Here we have collected raw read counts of 5 diagnoses versus 5 relapsed chemotherapy samples and 5 diagnoses versus 5 relapsed transplantation samples. Now then we proceed with the dimensionality reduction analysis using PCA after quantile normalization. The PCA were plotted between pre and post the intensive chemotherapy and pre and post transplantation. After studying the variance between these two, we went ahead with the differential gene expression analysis using DSEG2 pipeline from the tBioinfo server, and then proceeded with the enrichment and pathway analysis using GSEA, KEGG-GO, and human GAGE to study the pathways and the enriched or significantly regulated pathways and genes that are implicated in this treatment conditions that led to the relapse. Now coming to the results section, we will first look at the PCA analysis. Here for the chemotherapy diagnosis versus relapse, we can see 4 of the diagnosis samples in this part of the quadrant and 3 of the relapse samples in this part of the quadrant. In the case of diagnosis versus relapse transplantation studies, we can take 3 of the relapse over here and 4 of the diagnosis samples over here. But there's not a clear separation indicated in both these samples. And so we cannot say for certain because both the diagnosis and the relapse are in the disease condition itself. If we take a look at the differential gene expression study for chemotherapy, we can only see 5 sRNAs being upregulated. Here the red represents significant upregulation. These RNAs on further analysis show regulatory role in RNA biogenesis in eukaryotes, and this might be because of the increase in demand of cell proliferation of leukemia stem cells and hence hindering chemotherapy. In the case of differential gene expression analysis of post-transplantation, we can see a large number of differentially expressed genes that aid to adaptive immune response, cancer cell signaling pathways, regulating leukocyte activation and migration, T cell cytokine and effector cell production, all in all aiding to leukocyte cell proliferation and migration, and hence immune escape. Here we can see the post-chemotherapy and post-transplantation enriched pathways from the KEGG-GO pathway analysis. In the case of chemo post-relapse analysis, we can see the pathways are upregulated are mostly for which aids in migration, so it might have aided the myeloblasts to migrate from the bone marrow into the bloodstream. In the case of transplantation, we can see migration as well, and also some neuronal morphogenesis. And there also seems to be downregulation of pathways such as MHC class II protein complex, MHC protein complex, and MHC complex binding, peptide antigen binding, all in all, which aids in enabling to differentiate between the cell versus non-cell conditions. Here in the KEGG-GSEA pathway enrichment analysis, in the case of post-transplantation relapse, we can see an elevated level in the cancer cell progression pathways, which are HER2 signaling pathway, CAMP signaling pathway, TGF-beta signaling pathway, cytokine receptor interactions, which regulate or facilitate the interaction and the progression of cancer cells, and AML cell migration. In the case of post-chemotherapy, we can see that ribosome and ribosome biogenesis in eukaryotic pathways have been activated, and which was also observed in the differential gene expression analysis. Taking a look at the pathway signatures that are implicated in transplantation, we have neurological related pathways being downregulated, tumor progressive pathways being upregulated, and tumor suppressive pathways being upregulated as well. And these are the common pathways that are implicated in this study. Further analyzing the pathways using human GAGE analysis, in case of transplantation, we can see there is a downregulation in both the MHC1 pathway and downregulation in MHC2 pathways, where the green color indicates downregulation and the red color indicates upregulation. So we can see both of them are downregulated, and these are important for distinguishing the self cells from non-self cells. In this case, they also affect the CD cells and CD8 and CD4 cells from recognizing these self or non-self, and hence hinders the killing of target cells, producing cytokines, and activating other immune cells, all in all aiding to immune escape and subsequently relapse. This pathway was also mentioned in the original paper. Another significantly regulated pathway is the cell adhesion molecule pathway. Here we can see two sections of the pathway. One is the immune system and another one is the neural system. In the case of immune system, we saw as in the last slide that MHC1 and 2 are both downregulated, whose main end product leads to differentiating self from non-self cells. And these downregulation might have been the cause of no allograft rejection taking place. This might have helped. Then there's also T helper cell and T cytotoxic cell upregulation. Leukocyte trans endothelial migration genes were also upregulated, and these help in the transfer of these myeloid cells from the bone marrow into the bloodstream. These are all linked to cell-mediated adaptive immune response. In the case of neural system, we can see downregulated genes when some of these genes have the role in infiltrating the central nervous system, which may be the reason of neurological disorder being implicated, hence showing a connecting link of AML to neurological disorder. Now, to sum up our analysis, in acute myeloid leukemia was diagnosed and remission by transplantation and remission by intensive chemotherapy occurred. Now, after that, relapse after the transplantation occurred and relapse after the chemotherapy also occurred. So in case of a transplantation, there were regulated tumor progressive and suppressive pathways, and along with these, neurological disorder pathways and genes related were also downregulated. From this, we can conclude that in the case of transplantation, in the initial diagnosis stage, there might have been susceptible to neurological disorder because of immunocompromisation, and hence they were downregulated later after relapse. In case of chemotherapy, a differential gene expression analysis shows no significant dysregulation in pathways except for a particular upregulation in the ribosomal RNA synthesis. These, however, were interestingly downregulated in transplantation relapse. Now we can use this mRNA biogenesis pathway as a therapeutic target against this chemotherapy resistance, we might say. These are the references. Thank you.